Okay, let's dive right into this explainer. Today we're exploring the classic fertilization pathway in assisted reproduction. When most people hear IVF, their minds immediately jump to a scientist looking through a microscope, meticulously injecting a single sperm directly into an egg. But honestly, there's this entirely different, incredibly profound method that leans way more heavily on natural biology. We're talking about standard insemination, a process where the lab sets up the absolute perfect environment, but biology still has to take the lead. Here's a quick look at our agenda for today's journey. We'll start with controlled coexistence, then move right into setting the stage and tackle the biological gauntlet. After that, we'll cover the risks and realities and wrap things up by comparing IVF directly against ICSI. Section 1. Controlled coexistence, the philosophy of conventional IVF. To kick things off, let's make sure we're totally clear on what we mean here. Conventional IVF, or standard insemination, is basically the process of exposing retrieved oocytes, which is just the scientific term for eggs, to a prepared sperm population right there in a culture dish. The core idea is that fertilization is allowed to happen entirely without direct sperm injection. It's a highly active, beautifully engineered laboratory technique for sure, but it is assisted fertilization, not direct fertilization. The lab is essentially taking a step back to let natural biological processes do their thing. Now, what's really interesting about this slide is how it perfectly captures the underlying philosophy. Think of it as a moment in the lab, where the next step is not micromanipulation, but controlled coexistence. Standard IVF isn't just unstructured mixing. It's a profound shift away from manual, human intervention toward a really deep trust in biological capability. The lab makes the introductions, sure, but the sperm and the oocytes still have a ton of heavy lifting to do on their own. Moving on to Section 2, setting the stage, the architect's role. In conventional IVF, the embryologist is basically the architect of this whole process, and their very first architectural decision is all about the oocytes themselves. In this method, the oocytes are typically inseminated as cumulus oocyte complexes. What does that mean? Well, it means the cumulus cells, which are this natural protective cellular cloud that surrounds the egg, are left totally intact. The eggs aren't stripped bare before insemination. By keeping those cells around, the lab preserves a highly physiologic, totally natural interface for the sperm to navigate once they're introduced. So, how does this actually play out in the lab? The workflow is honestly incredibly precise. First, those cumulus oocyte complexes are placed into pre-equilibrated media inside a culture dish. That media, it's been carefully balanced well in advance. Next, they add a meticulously prepared sperm suspension. And finally, the dishes are quickly rushed back to an incubator for co-incubation. This environment is under ridiculously tight controls. We're talking heavily regulated temperature, pH stability, and specific gas mixtures, just to perfectly mimic the human body. The actual moment of insemination is super brief, but the setup is heavily planned. So the crucial point is the insemination dose. The specific concentration of sperm they add to that dish is definitely not just some random detail. It's a highly controllable, absolutely critical variable. The lab is aiming to provide just enough motile sperm to support fertilization. But here's the catch. If there are too few sperm, the chances of fertilization just plummet. On the flip side, if there are too many sperm, it significantly spikes the risk of abnormal fertilization. It is a very delicate microscopic balancing act. Let's move to and see how this builds in Section 3, the biological gauntlet. The arena. Okay, so the embryologist has essentially built the ultimate microscopic arena. But now we really have to ask ourselves, what must the sperm do entirely on their own? Because the second that dish goes into the incubator, human hands are completely out of the equation. There's this major myth out there that standard IVF is somehow the easy or passive route compared to direct injection. Well, this completely shatters that myth. The sperm have to conquer a rigorous, multi-step biological gauntlet. First up, they have to remain viable and independently swim just to reach the oocyte. Then they have to interact with those cumulus cells and successfully bind to the zona pellucida, which is the egg's tough outer shell. Third, they have to undergo the acrosome reaction, a highly complex cellular release that basically allows them to push forward. And finally, they must physically penetrate that shell, fuse with the membrane, and activate the oocyte to actually kick off fertilization. That is a massive undertaking. All of this means that conventional IVF requires the sperm to actually prove their functional competence. Because the lab isn't physically dropping a sperm inside the egg, standard insemination demands that the sperm complete that entire biological sequence of fertilization totally on their own. It tests the biology in a way that direct micromanipulation simply bypasses.
Section 4. Risks and Realities. When Biology Falters.
Naturally, because this permissive method relies so heavily on nature doing its thing, there are inherent risks involved. I mean, even when both gametes are placed perfectly in the dish, fertilization can still just fail, or the rates can be really low. Another major risk is polyspermy, which happens when more than one sperm enters the oocyte. And like we touched on earlier, this abnormal fertilization is heavily linked to having too high of a sperm concentration in the dish. Also, because the eggs aren't stripped bare at the start, the lab can't immediately confirm if they're mature. That means some immature M1 oocytes might be hanging around in there, rather than the ideal, fully mature M2 oocytes that are required for success.
Section 5. IVF versus ICSI. A Tale of Two Methods.
And this brilliantly illustrates the stark contrast between our two primary fertilization pathways. Looking at one side, you have conventional IVF. It's permissive, it's biology-driven. It demands full functional competence from the sperm, and that natural cumulus cloud is left totally intact. But on the other side, you have ICS, or intracytoplasmic sperm injection. ICS is highly interventionist. The cumulus cells are completely stripped away to check for maturity, and a single sperm is manually injected, bypassing almost all of those biological function barriers we just talked about. Neither one guarantees success, obviously, but they test and support fertilization in entirely different ways. Honestly, if you take away just one thing from this entire explainer, let it be this thought right here. Conventional IVF is where the lab creates the opportunity, but biology still has to close the deal. It's just such a beautiful, delicate partnership between meticulous laboratory science and the raw, incredibly complex power of human biology. Which leaves us with a final, really fascinating question to ponder. If conventional IVF demands so much more from biology, requiring the gametes to pass test after test entirely on their own, what does that reveal about the true nature of fertility that micromanipulation might actually hide? Think about that for a bit. Anyway, thank you so much for learning with me today. Keep asking those great questions, and I'll catch you in the next explainer.